Continuing on looking at specificity from the computational lens, next we have Professor Maria Rodriguez Martinez of Yale University. And Maria, are you with us?
Hi, hello, Justin. Yes, I'm here.
Hi, Grant. Please
Here.
take it away.
Let me share my screen. Sorry, this is the wrong slide here. Okay, so thank you very much for the invitation. It's a great pleasure to be here. I'm gonna tell you, so I moved recently to Yale, well, recently 1 year ago. So I decided to organize this talk to tell you a little bit about the work I was doing in my prior institution before I was at IBM Research in Switzerland. So I will spend some time to introduce that work. And then I want to be telling you about the work we are starting to do here at Yale. And this is work in progress. So I can tell you the direction, I can give you some glimpses of what we're starting to find, and I will be happy to discuss future directions. Okay, so let's get started. So starting with the work we're doing at IBM, my team for the last 4 to 5 years has been quite focused on T cells. I guess I don't need to make a big introduction about what are T cells, or the therapeutic potential. We all know that predicting binding specificity is critical for many biomedical applications, such as cancer immunotherapies. We are also in my team quite interested in autoimmune diseases, so we are also interested in identifying how different T cell receptors may be used for diagnostic markers of autoimmune diseases. So therapeutic applications are great. Now, because of this interest, of course, many people in recent years have been quite interested in developing computational models and machine learning models to predict binding specificity, my team also included. So we wrote this review last year. We were surveying works published until we put a cutoff only December 2023. And you can see there is a clear trend. The amount of work published is exponentially increasing. The trend has not slowed down. They also see some emerging paradigms emerging. So we start seeing in recent years, people start to use deep learning approaches and protein language models. Victor mentioned about it. We are also using them. Many people are using them. And although they have not resolved many of the standing challenges, certainly they have pushed even further the predicted capabilities of many of these models. Now, most of these models, until at least very recently, were based only on sequence. That means that, yes, we were focused on T cell receptor sequences, probably epitope sequences, maybe MHC, but the structure was completely ignored. We have seen as recent benchmarks, the one for example is organized by Justin and colleagues, have demonstrated we are plateauing. There is a limit to what you can predict only based on sequence. And in recent benchmark, actually, we have observed that the models that perform best, they were using some structural information. And even these models were having challenges to predict binding to unseen epitopes. Of course, there was the hope that generic structural models such as AlphaFold will resolve the problem, so we will be able to just do AlphaFold on everything and predict binding specificity. But even that, we are starting to see that AlphaFold has limited capabilities certainly for T cell receptors because of the flexibility of the CDR loops, also for antibodies. So I think the community is realizing we need to start rethinking how we're going to approach these challenges. So after saying that, let me take a few steps back. I'm going to tell you the work my team was doing at Zurich, focused on interpretable deep learning, and then I will move to the work we are starting to do at Yale. Okay, so when we started thinking about T cells a few years back, we're at the beginning of the wave of interest. We wanted to develop an interpretable deep learning model based on sequence that will integrate the sequence of the T cell receptor, the sequence of the epitope, and predict binding probability. Again, this is probably quite trivial for this audience, but let me just remind you again about the theoretical complexity of this task. And the challenge is that we don't have enough data. So when we were developing this model, again, a few years back, these numbers have increased by now. But back then, we have less than 50,000 receptors from public databases and less than 200 sequences of epitopes. By I mean sequences for which we have binding affinity. Of course, with immunopeptidomics, we have a lot of sequences, but we don't have the binding pairings. So for us, they are not useful for this type of modeling. So looking at these numbers, of course, we knew that trying to build a model that would predict binding specificity towards a new unseen epitope was impossible. You don't have sufficient data. So we focused on the other. Well, we built a generic model, but we knew in one direction the prediction was not possible. So we focused on the other direction. Just let me show you some results. This model, we developed a model called Titan. The model was trained on bulk sequencing, so by now it has been outperformed by models trained on single-cell sequencing and models using protein language models. But maybe what was interesting of this model was that we were specifically interested on the interpretability of the model. So we wanted not only to predict binding accuracy with good accuracy, but also to tell us which amino acids were important to predict the binding specificity. So for instance, to give you an example of the type of predictions that the model could do, these three CDR sequences. They are predicted to bind, well, they are experimentally measured to bind the same epitope. And of course, we don't have the ground truth. So we love to have the crystals and to look which amino acids are in contact. We don't have them. But at least we can do a sanity check. You can look that the amino acids that are predictive with high attention. So by the way, I guess most of you are familiar, but here with attention mechanism. So basically, dark color indicates that the model pays a lot of attention to this amino acid. And for us, attention is a surrogate of importance. We want to believe that if the model pays attention, this amino acid is important. Although there is a lot of debate about this. But for now, let's assume that if the model pays attention to this amino acid might be involved in binding. So as I was saying, the model at least identify conserved motifs across sequences. So at least this is not a validation. At least we can assume that these predictions make sense. It was also interesting to look at the attention pattern we got in the other direction. The one I just said from the beginning, it's not possible to predict. The one where you fix the T-cell receptor and you try to predict binding to different epitopes. Of course, this is an unsolved challenge, so we did not get a good performance, but it was good. It was interesting to look at the attention patterns. So basically, across all our epitope data set, we got the same pattern, that the model was looking always at the same positions with the same intensity, which was very strange. So it took us quite some time to figure out this because we first thought that there was a bug and we were kind of revising our code for a long time. And eventually, we realized that what the model is doing here is simply memorizing the data, which is rather smart. So we're asking the model to predict something that is not possible to predict with the amount of data that we have available. So the model just simply, okay, let me memorize the data. So at least for this set of epitopes, I will give you a perfect prediction. Of course, I cannot generalize outside of distribution, but this is a different question. So I'm showing these positive and negative examples of something where we could predict and something that we could not predict to just highlight once more why interpretability is critically important, especially in computational biology, where typically models are underpowered. So in the case where you have sufficient data, the model can tell you the interpretable approaches, and there are many, can tell you about which patterns or which features are more important. That can help you generate hypotheses to improve the prediction, to design new binders. But equally important, when your model is not predicting, as in the case of predicting binding to new epitopes, it can give you very clear hints that the model is overfitting or memorizing the data or just predicting something that doesn't make sense. And this is equally important. Okay, so maybe I would like to mention another work we did also in the area of interpretability before closing this chapter of the old work and moving to the new, very exciting work. So as I said, my team in Zurich was having for quite some time focus on interpretability. We have developed quite a lot of work and papers on interpretable deep learning, not only focused on protein sequences, but in the context of TCR binding, something that I did not like of attention layers. Attention layers is kind of easy and intuitive of apply. You plot, you integrate your attention layer in your model, and then you get these heat maps. The challenge with that is sometimes you can get these very difficult to read maps where you get this kind of diffuse attention, so the model is looking everywhere and you don't get a clear pattern. And these models, these attention patterns can be quite hard to interpret. So we wanted to develop, what we wanted to do was something a bit more, what we call human understandable. So that means for us humans, we work with if-then rules. That means if you give me this amino acid in this position and this amino acid in this position, and this amino acid in this position, then I'm going to predict this. So we wanted to have this kind of mathematical rule. So there are approaches to do that, and what we develop, we can show you one example of how can you develop this type of more understandable, interpretable model. So this is the example, they call it the code. It's an interpretable pipeline that we develop for TCR binding prediction models. It's model agnostic. That means that you can apply it on top of any black box model, so it doesn't need to know any information about the model that is behind the prediction. And just for the sake of explanation, we just develop a very simple test data where we focus on a single epitope. And we just develop a data set of TCRs divided into binders and non-binders. Then to reduce a little bit the computational complexity, we cluster the data because, of course, in the end, what we're going to do is to infer a rule. So that means an explanation per cluster. You could, of course, also infer an explanation per sample, but then you will end up with 10,000 explanations, which might not be so understandable. So instead, we cluster the data and focus on understanding the centroid of each cluster. Now, this is when the interpretability happens. And here, this pipeline is quite optimizable. So you can, we have also developed my own team models for interpreting TCR prediction models. So we could also change, it's possible to change the interpretability module and put your preferred model. For the sake of demonstration, here we use anchors, which is one of the most popular interpretable models. So anchors, again, is a model agnostic approach. It's just can be plugged to any black box model. What it does, at least at the conceptual level, is rather simple. So anchor focuses on identifying local explanations. Okay. So anchor focuses on unraveling local explanations. That means explanations that only apply to one single sample. So for instance, suppose that this is the latent space of your classifier, you are predicting binding or non-binding, and the blue region is binding, the pink region is non-binding. Typically, the task of a classifier is to infer this boundary decision. And in general, this is very complicated, especially with deep learning models. This boundary can be highly non-smooth and very high dimensional. So it's not something that you can do linear regression and separate the classes. So what anchors that is focusing on a real sample. It will just look at a neighborhood and then generate synthetic samples. And then you can do it as long as you have a black box model, you can just generate fictitious sequences and assign a fictitious label using your black box model. And then using the real and the synthetic sample, that you can infer if-then rules using a reinforcement learning approach. Just to show you a few examples, so we apply the code to our model Titan and another model that was published more or less at the same time, PMTNet. They were applied to the same data set, and I was surprised and not surprised in these results. So maybe let me first explain this plot. So here in the bottom row, we are just showing the logos of the particular data sets. So models were looking at different clusters, first surprise. So you see that the data sets, this is the frequentist representation of each cluster. And these are the rules on top of it. You can see that in many cases, the rules correspond to the most common amino acid, but not always. In other cases, for instance, here, the model, the code is choosing not the most frequent amino acid, but a different one. Also, the surprising, well, not surprising, but it always takes some time to think about it. Models, the rules inferred to each model were completely different. And we did many different runs, we did different explorations. We always found different rules. Again, this is for us humans, it goes against our idea of interpretability. We would like to believe that there's universal explanation. But this is not how machine learning models work. So machine learning models are typically over-parameterized. That means that they can reach the same correct conclusion in many different ways. So probably we have to start, it's the same way that you ask 10 different persons, explain to me what is a house. Everybody's going to come with 10 different explanations. Probably all of them are correct, but they are different. So pretty much the same happens with machine learning models. So that also opens a lot of philosophical discussions about how we evaluate explanations. We have done some work in this direction also. And basically, just to summarize an ongoing debate in kind of a few seconds, well, there's no universal way in which we can decide what is a good and a bad explanation. There are some general categories, such as simplicity. Explanations should be as simple as possible. And broadness, they should explain as many data points as possible. And of course, they should reproduce the data. Okay, and after saying that, I'm going to close this chapter on interpretability, and I want to still have a little bit of time to present the work we are doing now at Yale. So let's leave for the time being sequence on the side, and then move to structure and, more importantly, dynamics. Let me start by showing you these two T-cell receptors. So these two TCRs, they bind the same epitope. Well, first of all, I will ask you if you can spot the difference. I certainly cannot. Both T-cell receptors bind the same epitope with pretty much similar binding affinity. One is highly specific, the other one is highly cross-reactive. So why there's this stark difference between them? Okay, so one possibility will be, of course, to overlay the structure one on top of each other. So we can do that. You can see that, again, the framework regions are pretty much similar. I don't see major differences. You see some difference at the level of the loops. But again, remember, both TCRs bind the same epitope, and the contacts are pretty much conserved, and they bind with similar strength. So again, that makes us ask it, how can these TCRs that are so similar have such different binding profiles? And the answer becomes a bit more clear once we start looking at dynamics. And this here means doing empty simulations. So for this is an empty simulations of the TCR that is highly cross-reactive. So what we can see here, so here in pink, you see the epitope, and you can see once we put the epitope there, there is, look at this red loop. This is a loop of the CDR3 loop. There is this loop that starts receding and being pushed back. And this probably, again, this is work in progress, but this probably explains why the binding is not, it's very unstable, and this receptor can bind many different epitopes because the binding is less specific than in the other case. This is just an example. There has been many different works that point in the direction that we have to start looking. If we want to understand better binding specificity, we have to start looking at structure first, but also dynamics, because there is a lot of information at the dynamical level. Of course, in an ideal world, we have infinite computational resources, so we could do infinite amount of MD simulations. In practical world, MD simulations are computationally very intense, even with clusters and GPUs. So what we have started to do in my team, we are starting to look into generative AI. So generative AI, there is this new generation of deep learning models that can not only predict particular task or challenge, they can also be used to generate data. There are many different categories. Diffusion models have got a lot of attention, but there are many other categories. Diffusion models, for instance, the ones that were used, that had been used, had got a lot of attention because the ones what you can use to generate new images. So all these images now you see on the internet are completely unreal faces that look absolutely real, but they don't belong to any person on Earth. These are typically generated with diffusion models. So diffusion model is one category, but we also have flow matching, distributional graph formers, bioemo. We have a bunch of these generative models specifically designed for proteins. So the idea we are testing right now is whether we can use some of this model, of course, adapt them to T-cell receptors, but use them to allow us a more efficient sampling of the potential post-conformational poses of focusing on the CDR3 loops. So this is one example of this task. For instance, this is a T-cell receptor. Here, for the sake of comparison, the framework regions in green are kept constant. And what we are sampling is 5 different poses of the CDR3 loop. So you see here in different colors. These are the different poses. And the good thing is that you see a lot of variability. So obviously, we are sampled different things. You can see that they are significantly different. The bad news is that it took us 10 hours with GPUs to generate this image. So certainly this is not scalable. We cannot just scale this 1 million times. We don't have the computational power. So we are right now investigating other generative approaches such as flow matching, which is, it uses similar principles. It's based on denoising that may use sample protein structure from noise. But it uses the sampling is much more efficient. So it's computationally faster. So here. So this is, for instance, what we did using AlphaFlow. AlphaFlow is a new model that has been published not so long, well, maybe 1 year ago. It's an implementation of this flow matching for proteins. So what we did, maybe let me give you some explanation about this picture. So for this model, we have MD simulation. This is a TCR. Here we are modeling the unbound structure because to model the flexibility, we decided to forget about the epitope for the time being, if the problem is already hard enough, and eventually bring back the epitope at a later time. So again, right now we are focusing on the unbound T-cell receptor. We have MD simulations for this system, and this is the representation. The blue points represent the different poses that the MD simulation explore. Of course, here we are assuming that the MD simulations are the ground truth. We also know that there is noise in MD simulations, that the MD simulations might also have their biases, but we had to start somewhere. So for now, we are taking the MD as the ground truth. What we are overlaying on top of it are different runs of AlphaFlow. This generative model based on flow matching. And as you can see, that pretty much for all the three loops and for different runs, so we try all sorts of tricks that starting with different starting points, different outliers, different parameters. But you can see that in all cases, we are subsampling. This model is not able to explore sufficiently the large conformational space of the CDR3 loops. So we were a bit disappointed, but not completely surprised, because again, these models, flow matching is not built for flexible proteins, although the authors claim that it has some power in this potential, in this area, is not intrinsically built for flexible proteins. Okay, so moving forward, we are now also investigating a different generative model. Now we are looking at models developed recently by Microsoft, specifically distributional graph former. Without going into technicalities, these models are rather complex. These models, just without going into technicalities, they are trained to sample, to represent the thermodynamic state of proteins. So basically, what is better about these models is they don't assume that there is a single static conformation, as AlphaFold does, but assumes that there is an equilibrium distribution. They try to sample as much as possible this equilibrium distribution. So again, for this simulation, we have the blue points is the MD simulation that we take as ground truth, and we are overlaying on top of it the poses we infer with the distributional graph former. And you can see that now we are doing a bit better. We are sampling better, so at least the sampling is not so constrained as it was before. But still, we have mistakes in that some cases the model is sampling way, is exploring too much. So with generative models, there's always the trend between exploration. That sometimes you want the model to explore, but not to explore too much. So this, as I say, this is work in progress. Something that we are exploring right now is whether we can fine-tune this model or just adapt this model to first help us with the exploration part, but then to add mechanistic constraints so we can eliminate poses that we think they are not energetically possible. So basically use these models to sample diversity and then to have a filter that allow us to remove these poses that we think they are not realistic. But as I said, this is work in progress, so I hope I can give you some update in a few months. Maybe the last part I would like to mention before wrapping up is also some work we are doing on antibodies, also using structure. Here, we are not using dynamics anymore. We are focusing on this static structural representation. But let me tell you a few words about this. So one of the hopes that when all the AlphaFold models and similar structural models was published was that we could use AlphaFold to reproduce the structure of antibodies and T-cell receptors. We're starting to see quite quickly, and there has been a lot of work in this area, that actually both AlphaFold and similar models such as Chai1 and Boltz and all these structural models are not very good, neither with TCRs nor with antibodies. One of the challenges is that these models are trained to reproduce one static structure, and we know that both TCRs and antibodies have highly flexible domains that are not well represented. So the question we ask ourselves is how we can still use, because there is a lot of information on AlphaFold, so we know some domains are not highly accurate, but still there is a lot of accurate information. So the idea is not to discard everything, to take the parts that we think are more robust and maybe improve those that we think are less robust. So how can we do that? Well, so one idea we are exploring right now is just to transform this complex structure inferred with AlphaFold to transform it into a graph. So in a graph, basically, this represents the same antibody-antigen complex. Here we are using a RBD antigen as an example, but basically this is highly that applies to any antibody antigen. So to transform this structural graph in this structural structure into a graph, basically each node each node in the graph corresponds to one residue. And then edges represent distance. So everything that is within a given threshold, typically 5 angstroms, but this is something we can modulate, has an edge. So basically now we have gone from this 3D structure to a 2D structure. So what is the advantage of doing that? So the advantage is that we are simplifying this structure. The hope, and this is a hope, the hope is that we are eliminating variables that are highly noisy, such as specific geometric distances, angle, rotation, bonds. All these variables are highly informative, but also highly noisy. So if we get rid of these noisy variables, we hope that we get just with the bare graph structure that represents some ground truth. And in other words, that if we represent the more correct we can just get with the basic structural context and get rid of things that are more uncertain. We also did another modification. And of course, what we want to do this is eventually to predict binding affinity for an antigen and antibody. Now, we have seen that typically there have been also a lot of machine learning models that focus on predicting binding affinity between antibody and antigens. Typically, most computational models present this as an affinity regression task. That means just give me the antibody, give me the antigen, and I will predict the affinity. This is very hard for many different reasons. First of all, lack of data. We don't have sufficient data to train these models. But also, it's typically most data set that we have now publicly available is a collection of many different heterogeneous. Thank you. So most of the public data set that we have right now mix different experimental platforms. Some experiments measure IC50 values, some measure KD values, others measure even different properties that are used as proxies for affinity. So basically, we don't have a whole universal or large data set that just measure without biases the same biophysical quantity. That is one of the challenges to predict affinity. So what we did is just thought about it, okay, instead of trying to predict the exact quantitative affinity value, which right now is not possible, let's just ask the model to rank pairs. That means we give the model a pair antibody-antigen and a different antibody-antigen, and simply ask to predict which one has higher binding affinity. This, from a machine learning point of view, is an easier task, because now it's a binary classification, and models typically are better with limited data sets. We also put a threshold, so we don't ask to rank pairs that have small differences in binding affinity. So we put here a threshold M, and our threshold was for us 10. So we only ask the model to rank pairs that have at least a 10-fold difference in binding affinity. And our reasoning was that less a 10-fold binding affinity could just represent experimental biases and noise. So we don't want to confuse the model. Exactly, and then we tested that this setup is likely to reduce noise sensitivity and improve generalization. Let me show you some in silico preliminary results. So sorry for the ugly table. We have, of course, when we develop models, we first want to compare them with other computational models before start doing experimentally grounded comparisons. So we did these initial comparisons. So we compare what a model called Valley affinity. So we compare it to different models, some of them like simply protein language models such as CSM2, Antiverti, based on using both the ranking, as I described before, and also regression, where you try to predict the quantitative affinity. We also compare with a very recent model. This is MINT, has been published just a few months ago. MINT is a protein language model, but it's a hybrid because it also has information about protein-protein interactions. So in a sense, it's a bit more aware of interaction between proteins. And then we also compare it with other structural models such as AlphaFold 3. And because we already know that AlphaFold 3 by itself is not good enough, we did AlphaFold 3 and also Boltz 1, and then we improved them, fine-tuned them using energy-based models such as FoldX. For the particular task, so we did, for our benchmark, we separated our benchmark into what we call it unrelated. It's called like local perturbations, means that we had small amino acid changes close to the paratop. And what we call here unrelated means that we have large changes. So one of the things that was, well, just overall, our model value was outperforming most of the models with some exceptions here. We found that for some particular experiments, the language models were doing relatively well, but overall, our model was outperforming other models. We had a few surprises here in this benchmark. For instance, I was expecting, it has been debated whether naively I would have expected that once you have a structure of sufficient quality, predicting small changes close to the paratop or close to the epitope, also the antigen, so predicting small amino acid changes should be an easier task. And the logic is that because the model has seen some quality structure. This is not what we found. So overall, local perturbations are much harder to predict that quite different, that very far away perturbations. And the interpretation we are giving right now, but if I will be happy to hear it, you have a different interpretation. The interpretation we're giving right now is that actually, when we have small changes, we are pushing the model to the limit because we are asking models that in the end they are trained with inaccurate structural representations, with inaccurate protein language models, and we are asking them to predict high-grained details, and they are not able to do that. They are failing quite strongly. However, predicting large changes, this is probably easier. For that, we can tap into the power of protein language models, as was also mentioned before. They are able to recapitulate broad changes, although maybe not fine-grained changes.
Maria, just a 3-minute warning here.
Okay, so yes, with that, I would like to tell you that this is ongoing. Of course, this is an in silico result, and we are now thinking about applying to a use case focused on gastric cancer, but since it's an ongoing process, so I'm happy to discuss if you have questions. So with that, let me conclude. My team is quite interested in developing deep learning models and interpretable deep learning models for to model both T-cell receptors and antibodies. For T-cell receptors, I told you a little bit about the earlier work we did at Zurich using sequence-based models, both Titan and also the code, which is our approach for interpretability. More recently, we are starting to look at the structure, how to model the structure, or how to integrate the structural information both about T-cell receptors and B-cell receptors. We are starting, everybody knew, but we are starting to think about how to represent the dynamical landscape of T-cell receptors, how to model loop flexibility. That some of the approaches are promising, but they are computation intense, so certainly there's work to be done here. And I did not talk about it, but you're also thinking about how more efficiently fine-tune protein language models for specific tasks. And with that, I would like to thank the people who did this work, certainly the people at IBM and also the people of the growing team at Yale. And I'm happy to take your questions. Thank you.
Fantastic. Thanks for that, Maria. Maybe time for just one quick question, Simon.
You mentioned fold-X and combination with AlphaFold. So we used that combination too, and we saw a problem because in AlphaFold 3, there is not an energy minimization step compared to AlphaFold 2. So AlphaFold 3 produces a huge number of clashes sometimes that can distort your results very badly. And fold-X alone, because of the rigid backbone structure in the system, cannot resolve these clashes alone. So you have to be really careful with that, just as a small hint.
Okay. Yes, thank you. We've been thinking about that too. So this is some work we did, an initial benchmark. We're thinking also to develop a more thorough pathway where we start with AlphaFold or Chai1 or any other structural model. And then as you do, we just verified, we do some structural verification to make sure there's not, as you said, sterical clashes, and then just go into the energy minimization. So certainly, yes, there is work to do here in the optimization. In this particular case, there were no clashes. We just verified. But in general, we have to do it.
Great. Well, thank you very much again, Maria. Unfortunately, we have to move swiftly on to our next speaker.